Okay, I'm going to be voicing over some of my PowerPoints since I don't think that it's going to be very possible for me to do live classes for you to watch and interact with me. I just don't think the technology is going to be able to handle it. So I am going to go through these PowerPoint slides. I'm not going to have any time for editing or cleaning these things up, so it's going to be kind of quick and sloppy, but the information will be there. I will also probably record for each chapter a video of me at a whiteboard writing down the things I would normally write down when I give this lecture along with this. So I'm going to go through this. I will touch base on some of the definitions, but I don't want to spend a lot of time reading definitions to you. And I will use the pointer to try to explain processes as best I can. Again, I plan to do a second supplemental video for each of these that I do. So with that, let's just get started. Chapter 7, what's DNA? Basically, DNA stores the information that the cell needs to produce proteins. All of your DNA codes for proteins. It codes using little 3-letter groups called codons. These 3 letters represent various nucleotides. We've covered all of that in our organic molecules part of the course, so you can always go back there and look at those chapters for a little extra information. This is a 3-dimensional representation of DNA. It is twisted into a helix, and basically, here's a nucleotide. A nucleotide always has a phosphate group, always has a deoxyribose sugar, DNA, deoxyribose nucleic acid, and then it has this nitrogenous base. The nitrogenous bases always match up A to T. Adenine binds to thymine, guanine binds to cytosine. These nucleotides are joined together by hydrogen bonds across the 2 strands of DNA. DNA is double-stranded. That's very important to remember. Here are your hydrogen bonds. And if you look through here, you'll notice C equals G. C is always attached to a G. A T is always attached to an A. TA, TA, CG, CG. Okay. Now, the purpose of this chapter is to explain how proteins are made and how DNA uses how DNA builds proteins. There's a couple of phases. First, the DNA is the master copy. So we have to find part of a DNA molecule and figure out which one is basically a gene coding for a specific protein. Okay, so we take the DNA and we find the gene and we get an RNA molecule out of it. This is called transcription. So we copy just the part of the gene we need to make that tells us how to make a protein and turn it into messenger RNA. That occurs in the nucleus, and that is called transcription. Transcription means to copy. Then the messenger RNA leaves the nucleus and goes out into the cytoplasm where it is read by a ribosome. We talked about ribosomes in chapter 3. Basically, they take messenger RNA and translate it into proteins. Okay? That process is called translation. It reads the messenger RNA, which is a copy of DNA, and converts it into a protein. Okay, so these are our 2 steps, transcription, translation. Each one of these has several phases. There's initiation, elongation, and termination. Those are the 3 phases of each one of these, transcription and translation. Okay, so I'm going to do more on this in a little bit, but a gene is found on a DNA. Now, this strand of DNA is basically a chromosome. If you remember back when we did our mitosis, meiosis, we talked about chromosomes and how they're made up of 2 strands of DNA. So one side of that strand makes a gene loci or gene, as we commonly refer to it. It codes for a protein. Okay? There are 3 types of RNA. In the nucleus, when we transcribe the gene off of the DNA, we basically make a copy of it into messenger RNA. Messenger RNA is just that. It's a messenger. It takes a copy of the gene out to the ribosome. The ribosome reads the gene and makes a strand of amino acids that will eventually become the protein. The other types of RNA are ribosomal RNA. The ribosomal RNA literally makes up the ribosome, while the transfer RNA is a molecule that, as the ribosome reads a codon for an amino acid, the transfer RNA goes out and grabs that codon and attaches it to the strand in the specific order as explained and translated from the messenger RNA. Let's look at a couple differences between RNA and DNA, just to review. DNA is double-stranded. It has these 4 nucleotides: adenine, thymine, guanine, and cytosine. Guanine always matches up to cytosine, adenine to thymine. RNA is a single strand of nucleotides. And instead of thymine, you have uracil. If you ever see now you can see a single strand of DNA and you will know it's DNA because it will have thymine in it. If you look at a single strand of RNA, you will see the RNA and it will have uracil in it. So if we were to give you a list of letters that make up codons, and we'll talk more about codons here in a minute, the part of the DNA that codes for a protein is only on one side of the double strand, so we would only present that one side as the gene. If it has a T in it anywhere, then you're going to know that that's DNA. If it has a U in it, you're going to know it's RNA. That's what's important. So, transcription, translation. There are on YouTube, and I recommend you look some up, several important and good videos that explain transcription, because I'm going to go through it on the paper, on this PowerPoint, but it's not going to work as well as it would work if we had a moving video. Now here's a video that is embedded, and you have the link for it in your PowerPoint on eLearn.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
Okay, that video was, I don't know how well you could see or hear that, but you can find these videos like that on YouTube all day long. And some of them will go into far more detail than others. I'll go through it on paper on this PowerPoint, but I think it's easier to understand if you see the process moving. Again, the link to that particular video is in your PowerPoint that I gave you. It's right here attached to this button. You just have to hit Open Hyperlink when you open up the PowerPoint program. So let's try to step through this. Basically, first we're going to make the copy. Now the copy is going to find basically the start spot here in the DNA. We're going to have to split the DNA open, and this little section will be, okay, this is the gene. This is what's going to be copied into messenger RNA. It can be from this side or it can be from that side. Doesn't matter. And it is done by this enzyme right here, RNA polymerase. Polymerase, it basically is going to make a copy of one side or the other of the DNA strand, and it will copy those correctly, and then it will make a messenger RNA strand. So here we show it starting right here. Here's the DNA template, and it's starting to make copies, and it's attaching nucleotides to the messenger RNA strand. I can't stress at this point how important it is for you guys to spend a little time learning the definitions of the words for this lecture. Otherwise, this isn't going to be very helpful. So you're going to need to want to do that. So there's a place that will tell you where to start and a place that will tell you where to stop on this. Okay, here's a drawing I did to try to explain this process of copying from DNA over here to messenger RNA. Now, this is the part that's going to be copied. Each one of these letters represents a nucleotide. thymine, adenine, cytosine, and guanine. Each three-letter combination makes up a codon. A codon codes for a specific amino acid. So basically in the nucleus, here's my chromosome. I'm going to split apart the DNA strands, and I'm going to make a copy. Now, the copy in messenger RNA would be, this T would be an A. The A would be U because remember, there's no thymines in RNA. There's only uracil. C to G. There's one codon. It codons for methionine. It is also the start codon. This will be very important. This tells the ribosome it's time to start making a protein. The next one, TAA, will be converted into, the opposite of T is A, the opposite of A is T, but in messenger RNA, the opposite of A is U. So AUU, that codes for an amino acid called isoleucine. Then TCA, the opposite of A is T, the opposite of C is G, the opposite of A is T in DNA, but it's U in messenger RNA. So, and AGU codes for serine. UAA, this, again, A to U, T to A, T to A. This is my stop codon. Okay, so I've got a very, very small little gene here that's only going to have 3 amino acids in it. And then the stop codon tells the ribosome, okay, you're done. You don't have to add any more amino acids. Now, in real life, there aren't nearly, this is way too short. There would be literally, could be hundreds or thousands of amino acids that make up a protein. But I'm just doing this like this to kind of demonstrate how it is converted from DNA to messenger RNA. That's called transcription. Okay? So back to our original picture here. We've got this RNA polymerase enzyme. Remember, an enzyme always ends in ASE usually. There are some exemptions to that, but that ASE tells you it's an enzyme, and it basically does work. So this polymerase enzyme is going to read the DNA and add nucleotides to a strand of messenger RNA that matches or is the opposite of the DNA strand that it's copying. The DNA strand that it is copying, the part of the DNA strand that it's copying is, again, it's called the gene. So here we're going to start with initiation. First, the RNA polymerase enzyme has to find out where to start. Okay? It binds to this thing called primase. Primase is a promoter gene. It's the beginning of the gene. It unzips the DNA. There's an enzyme that does that. It splits it open. And then this polymerase starts copying nucleotides into a single strand. Now here it's started copying, and it's already got a strand of messenger RNA being formed, and you can see that it's matched up. The polymerase moves down the line of the gene in the DNA, making a single strand of messenger RNA. These are called complementary strands because it's a copy, an opposite copy of the original DNA. It continues until it hits termination. Now this previous phase, that was called elongation. So we did the initiation, we started, then the messenger RNA got longer and longer and longer, and then we finally hit the end and we got to terminate it. So we don't have to make any more. We have now made our messenger RNA strand. The primase comes off. The DNA resumes its original shape as a double-stranded molecule. And we have completed this messenger RNA. Okay? Now that we've completed this messenger RNA is going to leave the nucleus, because that's where our chromosomes are, and go to a ribosome, and the ribosome is going to start reading or translating that messenger RNA into a protein. Now, all of these nucleotides form these little 3-letter groupings called codons. Okay? Codons code for specific amino acids. So AAG is the codon that says lysine, UCA, serine. And you have an entire list of the codons you can use. You don't have to memorize these codons, okay? If you have questions on it, these questions will have this chart for you so that you can look it over and see what you need to see. So if there's questions on something like this, you would have a chart to look at. And basically what this shows the little codons that code for the various amino acids. Now, there are multiple codons that will code for the same amino acid. There's also this start codon, AUG. That probably wouldn't be a bad one to memorize. And then there are 3 different stop codons, UAA, UAG, and UGA. Okay? Wouldn't be a bad idea to remember what those are. Translation is going to build the protein.
A transfer RNA molecule is composed of about 75 nucleotides. Each transfer RNA molecule recognizes only one amino acid, which becomes bonded to the top of the transfer RNA molecule. Located on the bottom of the transfer RNA molecule are 3 nitrogen bases called an anticodon that pair up with messenger RNA codons during translation. As translation begins, the first codon of the messenger RNA strand attaches to a ribosome. Then, transfer RNA molecules, each carrying a specific amino acid, approach the ribosome. When the transfer RNA anticodon pairs with the messenger RNA codon, the 2 molecules join. Often, the first codon on messenger RNA is AUG, which codes for the amino acid methionine. AUG signals the start of protein synthesis. The messenger RNA then slides along the ribosome to the next codon, where a new transfer RNA molecule carrying an amino acid will pair with the messenger RNA codon. The 2 adjoining amino acids then become joined by a peptide bond, and the first transfer RNA molecule is released by the messenger RNA. As the process continues, a chain of amino acids is formed until the ribosome reaches a stop codon on the messenger RNA strand.
Now, there are numerous videos on this topic as well on YouTube, so if you don't like that one, you can find others. Let me move my... So, building the proteins, translation from messenger RNA to a string of amino acids that make a protein have the same 3 steps as transcription: the initiation, the elongation, and the termination. These happen at the ribosome. And what literally happens, initiation, basically a ribosome forms around an RNA molecule, and it reads the RNA molecule codon by codon. It looks for this little start codon, AUG, and that's what it starts, and that's codes for methionine. So a transfer RNA that has an anticodon to pick up this methionine comes to the ribosome and attaches it to start the amino acid strand. Now, all these amino acids are going to be attached together by peptide bonds. We covered that in the organic molecule section. The transfer RNAs hook up, bring the amino acids, and these amino acids form a covalent bond that is a peptide bond. Now here we can see transfer RNAs bringing these amino acids, and I'm going to pause this. Let me move it a little bit. This is the transfer RNA molecule. Here's one. It's got an amino acid here, and this is the anticodon. The transfer RNA molecule has the anticodon. Important safety tip, folks. And as it reads each codon on the messenger RNA. This is the messenger RNA strand. This is the ribosome. And it reads each one. It brings a transfer RNA, matches up its anticodon to the codon, bringing the correct amino acid to the ribosome to be attached by a peptide bond to the growing chain of amino acids. So this continues. There's methionine. Here's the anticodon to AUG. And this little line here is the peptide bond, and this is the string of amino acids being attached, and it just reads it, codon by codon, attaching each appropriate amino acid. They're linked, and eventually the protein is finished when it hits a stop codon. Here is an actual picture of a protein being built, and it's basically this one strand here is messenger RNA, and it has multiple ribosomes cruising along on it, and all of these little purple strands are strands of amino acids that will become proteins. Now, during this process, mutations can occur. Mutations occur when a copy of the DNA is not copied correctly. Mutations occur, the ones that get passed down and become part of the organism, come from the sex cells. If you have a mutation in a skin cell, you can't pass that to your offspring. But the mutation would have to occur in your sex cells. So if you're a male, the sperm, and if you're a female, the egg. And if we look back here at these little codons coding for different things, let's see, UCU or UUU, those are different amino acids. So one nucleotide gets out of place in a sex cell when it's copying your DNA, and you would have different proteins. And it doesn't take huge amounts of mutations to cause huge changes. A mutation in just one little gene, and I've done this in grad school, you actually could splice genes, and instead of growing the antennas, it will grow legs out of its head. Okay, these things, mutations don't have to be huge. The tiniest little mutations can cause massive differences. And I can give you some examples. Here is sickle-cell anemia, the disease in humans. Now, the normal gene has the codon for glutamate, and a sickle cell gene has the codon for valine. So let's go look at these, glutamate and valine. Glutamate, where is it? And valine, look at valine. Okay, so valine, GUU, GUC, GUA, GUG. And glutamate, glutamic acid, if you get a GAG instead of a GUG, you would make glutamate. That's normal. You get the GUG, you get the valine. So if you get a U stuck in there, that is an alteration. That is a change in your genetics. It just takes one nucleotide to alter the difference, and you'll get a valine instead of a glutamate. So just one nucleotide out of a codon of 3 in a sex cell passed down, and bingo, you've got the sickle cell disease. Now, why does sickle cell exist? It's an interesting malady. When you have a normal gene and a sickle cell gene, it makes you very resistant to malaria. And this gene first showed up in equatorial Africa, the malarial belt, and that's why it stayed in. It didn't get bred out. But when two heterozygotes come together, the odds of producing someone with two valines or 2 sickle cell genes, it's a lethal gene and can kill you very quickly. Here's a couple other types of mutations. You can have an insertion mutation. So you've got all these strands of nucleotides, and let's say you've got a chromosome with 40,000 codons. And in the 937th codon, you get an extra nucleotide stuck in there accidentally in the process of meiosis as they're copying it. Well, that's going to shift and alter every codon from that point on. So the entire, just adding one nucleotide would cause every codon after it to shift. Substitutions, instead of adding one or losing one, it just changes one. Like from GAG, which is glutamate, to GUG, which is valine. A frame shift can be a deletion or insertion in a sequence, shifts the way the sequence is read. Mutations can be good, they can be bad, or they can be neutral. And mutations can be good today but bad tomorrow. As our environment changes, mutations benefits can change. So what might be a benefit today might be detrimental tomorrow. And we know that these mutations occur during mitosis and meiosis, and that explains why we get these mutations. Now, here's some mutations that we have helped along either through selective breeding or gene splicing, things like seedless cotton. Most of our crops have been genetically modified. Like there's a lot of crops now that are immune to Roundup. And by being immune to Roundup, you can spray that herbicide on and kill the weeds, but it won't mess with the food crops. The downside to that is practically all of our crops are identical. They're genetically identical. So if a disease comes, there isn't going to be a lot of genetic variability, and that puts us at risk of losing an entire harvest. Okay? Mutations create different versions of alleles. Alleles are versions of genes. So you have a gene for that influences your hair color, how much melanins you produce. The different alleles would be the allele, the version of the gene that produces dark hair, and then there'd be a version of the gene that wouldn't produce melanin, and it would produce very light hair. Those are alleles. Viruses. Everybody's interested in viruses now. Viruses are usually just strands of DNA or strands of RNA. And we know that viruses also can get into our system and can rewire and recode some of our DNA. Now, in order for viruses to reproduce, they have to enter another cell. And here's kind of how that goes. Okay? Here is a virus. Okay, and it's just basically a strand of DNA or RNA that has a protein coat. It invades a cell. It attaches to the host and it invades the cell. It penetrates. It releases its DNA, which goes into the nucleus and hijacks the regular cell's DNA. Now, the host cell that has been taken over by the virus starts making copies of itself. And when it fills up this cell, then copies start going out and looking to infect other cells. Now, you have certain defenses. What will happen, a cell that gets infected with a virus will oftentimes put some of the surface proteins from the virus on the surface of its cell, so that when immune system cells come and interrogate that cell to see if it belongs in the body, is it part of self or not? We've talked about that in chapter 2. It will be identified as an infected cell, and that infected cell will be terminated by your immune system. Okay. These are the stages: attachment, penetration, synthesis, assembly, and release. Viruses can cause diseases in plants. Viruses aren't restricted to animals. Different groups of animals have different groups of viruses, and when you have a really bad pandemic or epidemic, that comes from the fact that viruses jump species, and jumping species is always a bad idea. Viruses are pretty species specific. Okay, for instance, we don't generally get infected by viruses from fish or plants or reptiles. Most of the viruses that jump species into us come from other mammals. Now, the best way to protect yourself from a virus, vaccines. Vaccines work very well. They basically expose your system to a version of the virus. And by version of the virus, all I mean is this surface protein on the virus. If you take some viruses and kill them and break them up and inject them into your body, then these surface proteins will get into your bloodstream and get inside your system, and your immune system will produce specific defenses that can produce an antibody that will attach to that surface protein and neutralize the cell so that it can invade a cell, and if it can't get in the cell, then it can't reproduce. And so that's what a vaccine is. They will take either a dead virus and inject it, or it's attenuated. By attenuated, I mean it has been treated with radiation, and the radiation keeps it from hijacking DNA inside the cell. It's inert. So it just floats around and stimulates an immune response. The immune response comes generally from B cells. B cells, these are immune cells, or several different types. One cell is called a B memory cell, and it is able to produce copies of itself, which will produce antibodies to render viruses ineffective. Prions. Prions are just proteins that are misshapen. They don't have the right three-dimensional shape. All proteins have a three-dimensional shape that allows it to function. If it folds improperly, it can cause serious problems. For instance, mad cow disease. Mad cow disease is caused by a diseased protein that gets into the brain. Its actual name is bovine spongiform encephalitis. And mad cow disease is spreading. It's on every continent now. When mad cow disease is discovered, they burn off the field, kill and burn the cows. I mean, they go to extremes to try to maintain the cattle herds, but pretty much the whole herd has to be sacrificed, and you can't eat the meat. And here is our list of vocabulary. DNA, that's deoxyribose nucleic acid. It's a series of A to T and C to G, adenine, alleles. Bacteriophages. Bacteriophages are viruses that infect bacteria. Let's see, we talked about codon, cytosine, elongation, envelope. The nuclear envelope, just as a reminder, that's the little membrane around the nucleus. We talked about these different mutations. Latent. Trying to see if there's anything here that... A mutagen. A mutagen is a substance that can cause mutations to occur in DNA. Let's see, homeotic gene, I don't think I talked about that. Homeotic gene is a gene that controls the structures of anatomical structures in the body. So you have the homeotic genes determine whether a snake has legs or not. Homeotic genes affect, You know, do you have a tail or not? Things like that. Do you have a fur coat? Those would be homeotic genes. This is all I have. If you have questions about this stuff, post it in the discussion boards that I've provided for you. And we're going to move on to the next video.